Hi, my name is James Greasley, coming to you from Vancouver, Canada. I'm excited to be here. In this first video, I'm going to walk you through a little bit about who I am, my background, and why I'm generally interested in this role. So first question, do I currently have a PhD? I do not, but I do have an advanced degree, a graduate degree in healthcare, and my healthcare training has given me a serious scientific foundation across a lot of different disciplines: anatomy, physiology, pathology, biochemistry, and not just surface level things. And honestly, I've never stopped learning. I'm a lifelong learner. I'm always searching out new information, new research, and so I believe that this makes me a well-rounded candidate for this role. So big question, why do I want to work for your company for Supreme Optimization? Well, when I first came across this role, it generally excited me. It really aligned with who I am. Healthcare and writing has always been two things that I really, really care about. And for a long time, they've always sort of lived in two different boxes. And something that I realized while working in a clinical setting is the impact that you can have on a patient is tremendous. I mean, it's incredible to help someone. It's very meaningful, but it also has a ceiling. And I've realized that good content doesn't have that ceiling. A well-written piece of content can reach thousands, hundreds of thousands, millions of people. It can help them make better decisions about their health, understand a treatment or a product, trust a company that's generally trying to help them. And I've seen firsthand that good content really educates, it builds trust, it moves people, and that is another form of helping people. It's just at a much larger scale. And that's exactly what Supreme Optimization does for its clients. So this isn't a role that just appeals for me. It's one that I generally feel aligned with, and I think I could do an amazing job based on my experience and my passion and what I really, really care about. So that's a little bit about who I am, why I'm here, and why I'm interested in the role. In the next video, I'll get into the science background and walk you through how I approach writing. Thank you for watching. Hello again. So this one, we're going to go over the actual work, covering a few of the science questions, and then I'm going to walk you through my approach to content from brief to finished piece. So let's get right into it. All right, so first question, what are competent cells? Who would use them? So competent cells are basically bacterial cells, usually E. coli, that have been prepared to accept foreign DNA. Normally, bacteria won't just let DNA in, so you have to treat them to make the membrane permeable enough. Now, once they're open, you can insert a plasmid, essentially a DNA construct that you've designed, and then the bacteria will replicate it for you. So suddenly, instead of a tiny amount of your genetic material, you have an unlimited supply. So typically, the people who would use these are molecular biologists. They use them constantly, cloning, drug development, protein production. It's a foundational tool that's used in a lab. Okay. Next one is, what is in situ hybridization? What is the use case? So ISH is a technique that lets you see where specific genes are being expressed directly within the tissue. The in situ part just means in place. You're not extracting anything. You're looking at the tissue as it is. You introduce a labeled probe that binds to your target sequence, and that label shows you exactly which cells or regions are active. The reason it's so useful is where not just in where this gene is expressed, but where in the tissue, which matters enormously in something like like cancer research. It's also widely used in developmental biology, tracking how gene expression changes as an organism develops. So for what would an isosome isolation kit be used? So they're used to isolate exosomes from biological samples like blood or urine so that you can study them. Exosomes carry biological information from the cells that release them, which makes them really useful for understanding diseases, detecting biomarkers, or developing new therapies. The kit gives you a reliable, standardized way to pull them out of a complex sample very cleanly. And big use cases right now are things like cancer treatments, cancer diagnostics, neuroscience research, and drug delivery development.